Hey everyone, and welcome to Miss Estruch Biology and this entire topic for theory video for A-level AQA Biology. And if you want for free a workbook to complete as you watch this, then check out the link in the description for exactly that. But if you want ready-made notes or flashcards which are mark scheme specific with examiners' tips that students have used to get them an A star, then don't forget to check those linked in the description as well. But that is it for now. Enjoy the video. AQA topic 4, and we begin with 3.4.1 DNA, genes and chromosomes. So comparing the DNA in a eukaryotic and prokaryotic cell is the first part of the spec, and this has been quite a common long answer question to do this comparison. So I've put it together here as a table so that you can see that direct comparison. And by the way, it's fine to present your answers as a table like this or a bullet point list in the exam, except for the essay. So eukaryotic DNA and prokaryotic DNA both are made up of DNA nucleotides. So that means they both have nucleotides that contain deoxyribose, the pentose sugar, phosphate group, and those nitrogenous organic bases. The nucleotides are joined together by phosphodiester bonds still, but the differences are eukaryotic DNA is longer than prokaryotic DNA. It's linear in shape, whereas it's circular for prokaryotic DNA, and then associated with the histones in eukaryotic DNA and not associated with histones in the prokaryotic DNA. So in eukaryotic cells, a DNA molecule is associated with the histones, forms a chromosome. So that's what we can see here. We've got the chromosome, and that is made up of tightly coiled DNA that is wrapped around these histone proteins. There is also DNA inside of the mitochondria and inside of chloroplast, which you learn about in topic 2. And the mitochondria within those organelles in eukaryotic cells contains DNA which is very similar in structure to the DNA that you get in prokaryotic cells. And the similarities are it's very short, it's circular, and it's not associated with proteins. So that is the properties of the DNA in mitochondria and chloroplasts, and that is a similarity to the DNA in prokaryotes. A gene is a base sequence of DNA, and it codes for the amino acid sequence of a polypeptide and functional RNA. And the mark scheme is really specific that you do say a base sequence of DNA, not just it's a section of DNA. And every gene occupies a fixed position, which means a certain place on a chromosome, and that's known as the locus. So for example, here we've got the locus for flower colour. The gene is for flower colour, that's the locus, meaning the location. You would always find it on the chromosome. A sequence of 3 DNA bases is a triplet, and that codes for a specific amino acid. And that is the fundamentals of the genetic code. There's 3 features of the genetic code that you need to know as well. So the genetic code is degenerate, it's universal, and it's non-overlapping. Degenerate then, this is based on the fact that there are 20 amino acids that the genetic code is able to code for. There are 4 DNA bases, guanine, cytosine, thymine, and adenine. And just a note here, although I've abbreviated it, you are not allowed to abbreviate those in the exam. You have to write the full name. And therefore, 3 bases are needed to make enough combinations to code for at least 20 amino acids. And we can prove this mathematically. So 4 to the power of n, there's 4 bases. N is how many of those bases you would need to get enough to code for 20. So if it was 1 base, that would be 4 to the power of 1, which is 4. So that would mean we'd only be able to code for 4 amino acids. Guanine would code for 1, cytosine would code for 1, thymine would 1, and adenine, and that's not sufficient. If it was 2, that would be 4 to the power of 2, and that gives us 16 combinations. So that is not enough to code for 20 amino acids. 4 to the power of 3 is 64, so that is sufficient to code for 20 different amino acids, plus there are extra. And that is what leads to this property of the genetic code being degenerate. Because there's 64 combinations, which is more than the 20 amino acids, as a result, each amino acid is coded for by more than 1 triplet of bases. So that is what we mean by the genetic code being degenerate. And we can see that here in this genetic code wheel, where you would start on the inside, work your way out. So G, U, C, that codes for Val, that's one of the amino acids. But GUU, GUA, GUC, all of those code for the same. By the way, this is a genetic wheel which always has the codons, which are mRNA. That's why we've got U for uracil instead of T for thymine. Now, it's an advantage that the genetic code is degenerate, because if a gene mutation occurs and it's a substitution mutation where one of the bases is now swapped and we have a different base, it might mean that the triplet of bases still codes for the same amino acid, and therefore even though there was a mutation, you still code for the same amino acid, so it has no effect on the overall protein that is made. Universal is the next feature, and this means that the same triplet of bases codes for the same amino acid in all organisms. So that is your definition. Now, this has been used to our advantage because we can do genetic modifications, because we know that you can take a gene from one organism, put it into the DNA of another, and because the genetic code is universal, it will still code for the same amino acids and make that protein. Non-overlapping means that each base in a gene is only part of one triplet of bases that codes for one amino acid. So therefore, each codon or triplet of bases is read as a discrete unit. So we can see here it's read as a discrete unit of 3, this is a discrete unit of 3, and so on. And that again is an advantage in case there is a gene mutation, because if one base does get substituted and it does code for another amino acid, at least it's only affecting one triplet, whereas if it was an overlapping code, that one mutation would affect 3 amino acids. So 2 other key concepts are knowing what coding and non-coding DNA is within your DNA. So there are non-coding, multiple repeats of bases that you find between genes. But in addition to that, within a gene, you do also have sections that code and sections that don't. So within a gene, only some sequences, and these are the exons, code for amino acid sequences. And you also have sequences within a gene that do not code for sequences of amino acids, and those are known as the introns. So these non-coding sequences, you do have them between genes, but also within genes. And it's the ones that are within genes that are called introns, and those get removed during one of the stages of protein synthesis that we'll have a look at shortly. So 3.4.2 is DNA and protein synthesis. Now, I've mentioned this term codon, but the definition of codon is 3 bases on mRNA that code for a specific amino acid. The start codon is a particular set of 3 bases, and it's the 3 bases at the start of every gene that initiates translation, which is the second stage of protein synthesis. The stop codon is 3 bases at the end of every gene, and this causes a ribosome to detach in translation, and therefore it stops translation. So it stops protein synthesis. Genome and proteome. The genome is an organism's complete set of DNA in a cell, whereas the proteome is the full range of proteins that a cell is able to produce. So the genome should stay the same. It's not going to change unless there are mutations. Whereas your proteome, that does stay the same because it's the full range of proteins, but the reason it says is able to produce is not every cell is going to make all of the proteins that it's able to, but the proteome is the full range that the cell is able to make. Doesn't mean it does make all of those proteins. So next, let's have a look at the 3 types of RNA. We have M, T, and R. mRNA is short, it's single-stranded, and you find it in the cytoplasm and the nucleus. It's made during transcription, which we're going to see shortly. That's the first stage of protein synthesis. And it's a copy of one gene. So it's going to be a complementary copy to that DNA sequence in the gene. And as we said, 3 bases on mRNA are a codon, and that codes for one amino acid. tRNA, or transfer RNA, this is found in the cytoplasm, and specific amino acids become attached to transfer RNA molecules. Each tRNA is specific for one amino acid. Each tRNA molecule has an anticodon, which is these 3 bases here, which are complementary to the bases in a particular codon. You also have an amino acid binding site at the top, which is where a specific amino acid will bind, and that's how tRNA can bring a particular amino acid. This is involved in translation. It brings the amino acids to the mRNA on the ribosome. It's still single-stranded, it's folded to create what's known as a clover leaf shape. And those bases, as they're folded, will form hydrogen bonds, and that's what holds it in this structure. Now, I did say we talk about rRNA. That actually mainly comes up in topic 1. rRNA, you just need to know that combines with proteins to make ribosomes. So let's move on to protein synthesis. This happens at the ribosome, but it does actually, the first step where you make mRNA happens in the nucleus. So we're going to go through transcription and translation. Transcription is when a complementary mRNA copy of one gene of the DNA is created in the nucleus. As we said, the mRNA is much shorter than DNA, and that's because it's only a copy of one gene rather than the entire genome. And because it's shorter, that means it's able to fit through the nuclear pores in the nuclear envelope. It moves out, and then it can attach to a ribosome in the cytoplasm to be used in the next step. So if you were asked to describe transcription, here are your key marking points for transcription. First of all, the hydrogen bonds between the DNA bases break, and that causes the DNA double helix to unwind and the 2 strands to separate. Only one of the DNA strands acts as a template though. You will then get 3 mRNA nucleotides from within the nucleus, attracted to and aligning opposite their complementary bases. And in RNA, the complementary bases, just bear in mind, because it's RNA for mRNA, you have uracil instead of thymine. So any place on DNA where there'd be adenine, uracil would be the complementary base pair. Now that's not normally a mark, point 4, if it's a describe transcription question, but it's just worth noting. The enzyme RNA polymerase is then responsible for catalyzing the condensation reaction, which joins the adjacent RNA nucleotides together, and in doing that, phosphodiester bonds are formed. Once that mRNA is copied, it then gets modified, and then it's ready to leave the nucleus. So these modifications then, we actually call the mRNA before it's been modified pre-mRNA. Once it's been modified, then we call it mRNA. So the modification that happens is, once you've made your mRNA, and we've now got pre-mRNA, the introns get removed, and that's known as splicing. So the introns are spliced out, which just means they're cut out and removed. So then you're just left with the exons, and the exons are the sequences of bases on mRNA that code for sequences of amino acids. Now that happens in eukaryotic cells. It won't happen in prokaryotic cells because prokaryotes, such as bacteria, they don't have introns in their DNA, so they don't have to go through that step. The next stage is translation, and this is when the mRNA has now been modified, it leaves the nucleus, and it's going to attach to the ribosome, and tRNA is involved as well. So here are our key steps. Number 1, I've just said. Number 2, the ribosome, once that mRNA is attached to the ribosome, will start at the start codon, and then it moves along the mRNA. But the key thing here is knowing that the mRNA attaches to the ribosome. Sometimes there's an extra mark for saying at the start codon, or it will move to the start codon. Mark number 3, always going to be a mark. A molecule of tRNA has an anticodon complementary to the codon on mRNA, and that's due to complementary base pairing. Number 4 is always a mark. Each tRNA molecule carries a specific amino acid, and that is determined by the anticodon. The ribosomes will then join those amino acids, and a peptide bond forms between the amino acids. ATP is required for the formation of that peptide bond, but it's actually also required to join the amino acid to the tRNA molecule. The ribosome will then move along the mRNA to the next codon. The process repeats with new tRNA molecules bringing more amino acids and binding them. And this will continue until a stop codon is reached. So your absolute key marking points that you should always include would be number 1, number 3, 4, 5, 6, and 7. So 1, 2, 3, 4, 5, 6. This is often a 4 to 6 mark question. So they might be, here's your range, you have to say 4 of those 6, or if it was a 6 marker, you'd have to say all of them. Next then we move on to 3.4.3, genetic diversity, and this is looking at mutations and meiosis. So genetic variation can be introduced in these 3 ways: through meiosis, mutations, and random fertilization of gametes. So meiosis is the type of cell division that creates genetically different cells, which are the gametes, so sperm and egg cells. And unlike mitosis, there are 2 rounds of division. In meiosis 1, that is when homologous chromosomes are separated. In meiosis 2, it's when the centromere splits and the sister chromatids are separated to the opposite poles. And by the end of both divisions, you have 4 cells. They are haploid, meaning only 1 copy of each chromosome, and they are genetically different. So I've just put this down here as a reminder of what haploid and diploid means. Haploid means 1 copy of each chromosome, and that is what you'd have in the gamete. So for example, the haploid number for a human is 23. Diploid means 2 copies of every chromosome. So for humans, the diploid number is 46. Now, the way that meiosis introduces genetic variation is the key focus here. You don't have to know PMATs, prophase, metaphase, anaphase, telophase, like you did for mitosis. You just need to know for meiosis how the genetic variation is introduced. And that occurs in 2 processes: independent segregation of the homologous chromosomes and crossing over between homologous chromosomes. You have to say homologous chromosomes for both of those. And if you're not sure what that means, homologous chromosomes are 2 chromosomes that have the same genes. So for example here, these 2 are homologous. They are the same size, same shape, and it's because they contain the same genes. You just have 2 copies of them. One that would have come from the mother, one from the father. Now, both of these processes here occur in meiosis 1, which is the first round of division. So let's go through independent segregation then. As we said, it happens in meiosis 1, and that is when the homologous pairs of chromosomes line up opposite each other at the center of the cell, or the equator of the cell. But it's random which side of the equator the paternal and the maternal chromosomes will line up. They're always in their pairs, but it's random which side they go onto. And that matters because when the homologous pairs get separated, those chromosomes are pulled apart to the opposite end, depending which side of the equator they're on, will determine which chromosomes you get in these cells here, and then those get separated, the chromatids. So that is how the genetic variation is introduced. And there is actually a maths question linked to this quite often, using this formula here, 2 to the power of n, because you can use this to work out how many possible different ways are there for these homologous pairs to line up at the equator, and therefore how many different gametes could be made. And it's 2 to the power of n, 2 because we have pairs of chromosomes, n is for the number of homologous pairs you have. So for humans, that would be 2 to the power of 23, which is over 8 million. So because of independent segregation, There's over 8 million different ways that our homologous pairs of chromosomes can line up at the equator, and therefore over 8 million different potential gametes one human can make, just from independent segregation. But on top of that, crossing over can also occur. Now, crossing over is rare. That becomes important when you get to topic 7, learning about autosomal linkage. But for now, we're just going to go through what crossing over is and explain how it introduces genetic variation. So it also happens in meiosis 1. Again, it's linked to this homologous pair lining up. So we get the homologous pairs lining up at the equator. And when you have a pair next to each other at the equator like this, we call it a bivalent. So they line up and they form a bivalent. The non-sister chromatids, one chromatid is one of these arms of a chromosome. Non-sister means it's the chromatids from different chromosomes. So the blue and the pink here are non-sister chromatids. They cross over and entangle, and the exact point where they cross is called a chiasmata. Chiasma is the singular, chiasmata is the plural. And at each of those chiasma, you can get a break happening, and then you exchange that section of the non-sister chromatids. And as a result, when these chromatids separate to go on to make the final 4 gametes from one round of meiosis, you end up with new combinations of alleles in the chromatids because they've swapped sections of the chromatid. So if we just compare meiosis to mitosis, meiosis is 2 nuclear divisions versus 1. Meiosis, you get a haploid cell versus diploid. Meiosis introduces genetic variation. Mitosis, you get genetically identical cells. You could also be asked for meiosis to identify where meiosis is happening within an unfamiliar life cycle. And the key thing here is don't look for where gametes are made, because for some organisms, meiosis isn't making gametes. So instead, what you need to look for is where you go from having a diploid cell to a haploid cell. And they might write diploid or haploid, or they might write 2N, meaning diploid, or N. So that's what you need to look for on a diagram. And this is a bit blurry, but we can see here that we've got N, 2N, N, N, N. So where we go from the zygote to the zoospore, we've got 2N to N. That would be the meiosis stage because we've gone from diploid to haploid. Not here where we have that gamete being made. Now in this one, we can see everything on this side is 2N, everything on that side is N. So that means at this stage here, it is, it's labelled on, but if it wasn't, this is meiosis because we've gone from having a 2N cell to all of these being N. So that's what to look for. Diploid to haploid is always where the meiosis is occurring in a cycle. Gene mutations is another way that genetic variation is introduced. And a gene mutation is a mutation in a gene, but it means you're changing the base sequence of the DNA for that gene. And these mutations randomly occur during DNA replication. So there's a link here to DNA replication in topic 1, and there's a link here to the cell cycle in topic 2, because DNA replication happens in interphase during S phase. So these random mutations are more likely to occur if you are exposed to mutagenic agents, and they can interfere with DNA replication, and that's why. So mutagenic agents include high energy radiation such as UV light, ionizing radiation, which would be gamma rays and X-rays, and chemicals, so carcinogens such as mustard gas and cigarette smoke. There are 2 types of gene mutation that you need to know for topic 4. You do learn more in topic 8, but for paper 1 or for the AS, you only need to know deletion and substitution mutations, which is modelled here. So here is our original unmutated DNA sequence. A substitution is when one base is changed, and we can see that cytosine base in the original has been changed to adenine, and maybe that might still code for the same amino acid because the genetic code is degenerate, or maybe not. A base mutation that is a deletion is when one entire base is deleted, and we can see that one of the cytosines has been deleted here, and all of the bases after the point of that mutation have now shifted left. And this is known as a frame shift because all of the codons after the point of mutation have now changed. So that means we're going to code for all of those, probably different amino acids. So a deletion mutation is far more harmful, potentially, than a substitution mutation because more amino acids will be coded for incorrectly. You can also get chromosome mutations, and this is mutations in the number of chromosomes, and that can happen during meiosis. Now, a chromosome mutation, if you're asked in the exam to name the chromosome mutation, it's non-disjunction. That's come up quite a lot recently, and sometimes students aren't sure what they're meant to say the name of the chromosome mutation is, and the name of it is non-disjunction. So non-disjunction, what this means is during meiosis, either the homologous chromosomes in meiosis 1 don't separate equally, or if it's meiosis 2, the sister chromatids don't separate equally. So as a result, you end up with more either chromosomes or chromatids going to one cell, and the other cell has a lack of chromosomes. So that is the non-disjunction chromosome mutation. Now this is just going to show you it, so you can visibly see the outcome. So in this scenario, it's in meiosis 1. The homologous pairs have lined up at the equator, and for some reason the spindle fibres are all coming from one of the centrioles, and that means all of those homologous pairs are going to be pulled to this cell. This one has got none, so that cell isn't going to survive, and this one has now got an extra copy of every chromosome in it. Meiosis 2 is when the sister chromatids separate, but because we had 2 copies of every chromosome, we end up with 2 copies in our gametes. So we have diploid gametes here instead of haploid. So this is one example of non-disjunction where we now have an extra copy of every chromosome in that gamete. Now, if that gamete was to fuse with a haploid gamete where there are no mutations, then 2 plus 1 equals 3. So we now have a triploid cell. So this is one example of non-disjunction. And this does happen quite commonly in plants, and plants can still grow and survive that, but this isn't seen in humans. Triploid would be fatal. That wouldn't lead to a fetus and a baby that could actually be born. Now, another type of non-disjunction we can see here, it's still happening due to the centrioles only producing spindle fibres on one side, but meiosis 1 happened normally. This time it's meiosis 2, where the sister chromatids are both pulled to the same side. This one, there was no chromosome mutation, no non-disjunction, but in meiosis 2, this one there was. All of the sister chromatids moved to one side, so again we end up with that diploid gamete and one gamete with no chromosomes. Now, another type of non-disjunction that can happen is if only one of the chromosomes, the spindle fibres, are attaching at one side. And as a result, both copies of only that chromosome move to the same cell. So we still end up with a haploid gamete, but you have one extra chromosome. So it's haploid, we've got one copy of all the chromosomes, except for this one, we've got one extra. And this one's haploid, one copy of all of the chromosomes, except we're missing one. So it'd be described as n plus 1, and these are n minus 1. And if you then had a gamete like this, combining with a haploid gamete, you end up with a diploid zygote plus one extra chromosome. And that's actually what happens in Down syndrome. There are 3 copies of chromosome 21 because of this type of non-disjunction chromosome mutation. If it was the n-1 gamete, then you'd end up with a zygote that was still diploid, minus 1 chromosome. Next then we move on to 3.4.4, genetic diversity and adaptation. And genetic diversity is the number of different alleles of genes in a population. And this is one of the key driving factors of natural selection, because you have to have genetic diversity in order to select which allele is going to be passed on. So a gene pool can be described as all of the genes and all of the alleles in a population at a particular time. The allele frequency is the proportion of organisms within a population carrying a particular allele. We're going to see these key terms come up as we go through natural selection and evolution. So natural selection is the process that leads to evolution. Your definition of evolution is the change in allele frequency over many generations in a population. And this helps to ensure that you have a population of species better adapted to their environment. And these adaptations could be anatomical, physiological, or even behavioural adaptations. So natural selection is random mutations occur. That's always your first marking point. If it's an applied example, you have to state which allele has been made. So in this case, it might have been there was a random mutation that created the white mouse or the black mouse, whichever one it was. That introduces genetic variation. Those are often a combined mark. Some mutations might be harmful, but some are actually advantageous. So if an individual did have that allele, they might be more likely to survive. And if they're surviving, they're more likely to reproduce. And if they reproduce, then they're passing on that new mutated allele to their offspring. And if this repeats over many generations, most of the population or a lot of the population will now have this allele in their DNA. So that means the frequency of that allele has changed over many generations, which was our definition of evolution. Now, there's two different outcomes that you learn about, which is types of selection or outcomes. You do learn a third type in topic 7, but there's only two that you need to know for paper 1 or for your AS. And one of those is directional selection. And this is when the allele that is providing the selective advantage is one of the extreme traits. So it could be very, very dark fur if it was the mice, or very, very light fur if it was the mice. Or the example that you actually need to know is antibiotic resistance. So bacteria that are very, very, very resistant compared to not very resistant or not resistant at all, the very resistant ones are surviving. Their alleles are then being passed on. And the outcome is we see this shift from the original red line showing us the original distribution of the different alleles in the population. Now we can see there's been a change in the allele frequency because most individuals now have that extreme trait. Stabilizing selection is if there's no change in the environment. So whatever was the most common or the modal trait, that is still the allele that's providing the selective advantage, and it still therefore remains the most common. It's just over many generations of natural selection happening, there's still a change in the allele frequency because now even more individuals have that modal trait and the extreme traits have been lost. So in terms of standard deviation, the spread of data around your mean, we've lost those extremes, so the standard deviation has decreased. The example linked to this is human birth weight. So a middling birth weight is the selective advantage because if you're much, much lighter, then potentially you were premature, and that means maybe you're less likely to survive. And if it's a very, very big baby, you're more likely to have complications with the birth. So the middling birth weight is the selective advantage. Next then we've got 3.4.5, species and taxonomy. The definition of a species is two organisms that are able to breed and make fertile offspring. But the key thing is, can they breed and make fertile offspring? If they can, they're part of the same species. And species must reproduce and pass on advantageous alleles in order for the entire species to survive. And courtship behaviour, we did say behaviour is an adaptation. Courtship behaviour is genetically coded for, it is an adaptation, and it is essential for successful mating and species recognition to help the entire species survive. So what courtship behaviour means is, it's the different behaviours that animals demonstrate as a way to attract a mate. And each species has their own unique sequence of behaviours to ensure successful mating. And what we mean by successful mating is if the two individuals are mating and produce offspring. Now, normally the courtship ritual is performed by the male, and then the female selects which male they want to reproduce with. And the Behaviours are often a sequence of behaviours. And it's not necessarily just behaviours, it could be sounds, the release of pheromones, which are chemicals. So it could be dance moves, displays of colour and feathers. There's lots of really extravagant courtship behaviours in plants. And in fact, if you just search birds of paradise courtship behaviours, there's some really cool David Attenborough videos on YouTube for that. So females will observe the courtship ritual and decide if they want to mate with that male. So the reason this is important is, as we said, it's to ensure successful reproduction, which means mating and producing offspring so that the whole species can survive. And the reason it increases the likelihood of an offspring being produced is it enables you, first of all, to recognise members of the same species. Now that sounds fairly obvious and not needed if you're thinking about humans, but for other animals, they do rely on this to make sure they are the member of the same species before they reproduce. Secondly, in order to be able to make offspring, they'd need to be mating with a member of the opposite sex. So that's the first two things that courtship helps animals to do. Recognise members of the same species, recognise the opposite sex. Next is synchronising mating behaviour. Now what this means is animals will do the courtship ritual and they'll be able to recognise what that ritual means only if they are producing gametes. So males will only do the dance when they are producing gametes. Females will only recognise and respond to the courtship ritual when they are releasing eggs. And again, that then means if they reproduce, you're more likely to have offspring because both the male and female are producing gametes, which could then fuse together to make an offspring. Now, on top of that, this isn't to ensure successful mating because this is now saying for some animals, there's an extra importance to courtship behaviour, and that's to ensure the survival of their offspring. And that's because these courtship rituals can help to form a pair bond, which means the two parents should stay together. And we see this in penguins because it's a massive advantage to the survival of the offspring if they have a mother and a father, because one of them stays to look after the young while the other goes to find food. It also helps to enable the female to select a healthy male to mate with, and therefore their offspring is more likely to be healthy and therefore increases the survival of their offspring. And the logic behind this is if a male is fit and healthy, then they'll have more energy that they can put into their dance routine, or their feathers will look brighter in colour, they might look better conditioned. So that is the idea behind that one. Next then is the binomial system. Bi meaning two, nomial meaning name, so the two name system. And this is when you use the genus and the species to name the organism. And this is the universally used, meaning it's the same across the world. The universal scientific way to identify an organism is the binomial system, genus and species. And this is useful because, first of all, you can share data, but also it gives you more information than the common name would. So, for example, we've got two birds here. The common name is New Zealand robin, and then this is a robin. And you might think, oh, they're both called robins, so they must be closely related. But if we have a look at their name using the binomial system, their genus and species is different. So they're not actually even closely related. So it's useful to be able to see, are different species part of the same genus? Because if they are, that means they're closely related. If they're not, then they're not closely related. And that links us into phylogenetic classification. And this is arranging species into groups according to their evolutionary origins and relationships. So it's a way for us to see for different species how closely related they are. Do they share recent common ancestors? And that's what these phylogenetic trees show. So humans, for example, in this tree, we're most closely related to chimpanzees because we share the most recent common ancestor. Then as we go back in time, so we're going back in time this way, after that, here is the next most recent common ancestor. So that means after chimpanzees, we're most closely related to these two. And then we can go back again and see it's then these, and so on and so on. So every fork or branching point represents a common ancestor, which means that is the species, anything that branches off, evolved from. Now, linking to classification still, individuals can be classified according to their physical and biochemical similarities, and classification systems are an example of a hierarchy. And that is a really common 2-mark question. What is meant by hierarchy? And it's when you have smaller groups arranged with larger groups. That's mark 1. Mark 2 is there's no overlap between the groups. So what that means is we've got an example of a hierarchy here, which you're probably familiar with from GCSE, and you do still need to know this for A-level. We can see that within a genus, there are lots of smaller groups. And that's because in one genus, there'll be lots of different species, but there's no overlap between those species. They're all different species, they just all belong to the same genus. And then we can go up again and say the same thing with genus. Within a family, there's lots of different genuses, but there's no overlap between them. 3.4.6 is biodiversity within a community. And biodiversity refers to the variety of life, and it can be measured on different scales. It can relate to a small local habitat, such as a pond or a woodland, or the entire earth. And there's different terms to classify or to describe biodiversity. We've got species richness. That definition is the number of different species present in a community. So it just tells you how many different species there are. Index of diversity gives you more information than that because it's looking at the relationship between the number of species, so the species richness, but it also takes into account the number of individuals within each species, which is far more helpful because then we can get an idea of, are any of those species at risk of going extinct, because we now know the number of individuals within each species. And biodiversities can be used to describe, as we said, local habitats or the entire earth. And a low biodiversity isn't always a cause for concern because it might be naturally there is just a very, very low biodiversity because there's such harsh abiotic factors, say for example in the Arctic or anywhere with extreme abiotic factors. The concern is when you have an area that had a high biodiversity and it's starting to decrease because something is causing individuals and maybe entire species to die out. And often that is due to human activity. Now we can calculate this index of diversity using this formula. They give you this formula, but they don't always tell you what the capital N and lowercase n is. Often they do, but I'd still say learn it because the most common mistake students make on this is misremembering what the lowercase n actually means. So capital N is the total number of every organism alive in that area, whereas lowercase n is just a population size of one species. The best way, though, to get an idea of this is we'll go through an example. Now, with this formula, typically, you will get a value that is between 1 and 10. It doesn't have to be 10 as the maximum, but usually you get an answer somewhere between 1 and 10. So if you do this in the exam using this formula and you get an answer like in the 50s or 100s, you've gone very wrong somewhere. Recalculate it, try again. So to model this then, capital N we said is the total number of organisms. So that is the sum of that column. We've got 4 species, added all the individuals together. Lowercase n is the population size of each species. And we have to do n times n minus 1, and then it's the sum of that for every species. So n is 6, so we need to do 6 times 6 minus 1, which is 5. 6 times 5 is 30. Then for this one, 3 times 3 minus 1, so 3 times 2 is 6. 12 times 11, 132. 4 times 3 is 12. And then the denominator is the sum of all of those. So you do have to work this out for every single species they give you in the table. Add that all together and that gives us 180. Then the numerator is the total number of individuals, which in this case is 25. So we do 25 times 25 minus 1, which is 25 times 4. And then that comes to 3.3 when we finish the rest of that equation. Farming is one of the examples of human activity that can reduce biodiversity. And within the specification, it says you need to be awareness of the balance of we need farming to provide food for humans, but there needs to be a balance between having that farming and not having too negative an impact on the biodiversity. So some of the ways that modern farming techniques might reduce the biodiversity are the removal of habitats. So that could be removing hedges and putting in fences instead, or just not having a hedge there at all. It could be removing habitats by cutting down a forest and then just planting crops. It could be using a monoculture, which means just growing one species. And if you've removed a whole range of plant species and replaced them with only one, reduce the species richness, but also the variety of food available for herbivores. If pesticides and herbicides are used, that is going to kill animals and plants, which again is reducing the species richness. And the overuse of fertilizers can cause harm in aquatic ecosystems as well. But two key marking points that always come up linked to this concept of farming techniques and how this can reduce biodiversity is there will be fewer habitats. So fewer habitats and fewer food sources. Not quantity of food, it's food sources, meaning one plant might eat grass, another plant might eat daisies. I'm just doing two random examples. So if you only have grass now, any of the animals that ate daisies can't survive there. So it's fewer habitats, fewer food sources if you have fewer plant species present. Next then, investigating diversity. So genetic diversity refers to the variety of alleles within or between species. And you can assess this by comparing the frequency of measurable or observable characteristics. So for example, the flower colour. But this can be less reliable because characteristics are also influenced by the environment as well as the genetics. So more accurate ways to compare are comparing the DNA base sequence. This is a direct measure of how similar or how different your DNA is. You could also do base sequence of mRNA because that is copied from the DNA. Or it could even be the amino acid sequence in proteins because amino acids are coded for by DNA. However, we do need to bear in mind for this one, because the genetic code is degenerate, two organisms might have the same amino acid sequence for a particular protein, but their DNA base sequence might be different because the genetic code is degenerate. So the most accurate measures are comparing DNA base sequences or mRNA base sequences. So when measuring variation within a population, this is where we get the concept of sampling, because we'd take samples because it'd be too time consuming to literally measure it for every individual organism, and it might not be very accurate. So to investigate variation scientifically by doing sampling, you have to make sure that you're doing random sampling to avoid bias. You must take large samples to ensure the results are representative, and then calculate a mean to summarise the data. You can then also go on to do standard deviation and even statistics. That is going to be the most useful because if you do a statistic, you can see if you have a significant correlation or a significant difference depending on what sort of pattern it was you were looking for. So that takes us to the end of topic 4. Hopefully you found it helpful. If you do want even more help with mark scheme specific key points, or if you want to have examiner tips, essay links, recall questions, then go for my A-level notes, flashcards. That is all about mark scheme key points, learning those terms. So I'll link those below and you can click the QR code or scan the QR code at the top. That's it though for today's video. Hope you found it helpful and I'll see you in a video very soon.